Hello, and thank you for joining Cure Alzheimer's Fund today for this important presentation. I am Meg Smith, CEO of our organization, and I hope you will allow me just a few minutes to introduce CureAlz. We were founded with one mission: to fund research that will accelerate prevention, treatment, and cure for this disease. To that end, since 2004, we have deployed more than $200 million toward extraordinary research pursued by brilliant and determined scientists around the world. Our dedicated donors make all of this possible, people who share our drive and commitment to end this disease and its terrible impact on patients, families, and our communities. We are grateful for their trust in us, and we are determined to honor it by finding and funding the vital research that is the only way to achieve progress. Our board of directors and trustees hold us accountable to our mission. They and certain other supporters cover all of our operational overhead. This generosity and vision mean that our other wonderful donors know that 100% of their gift goes directly to our research program. At CureAlz, our only goal is to find solutions to help the Alzheimer's community. We do not pursue profits or financial returns. All of our scientific decisions are made by active researchers who themselves are at the forefront of discovery. Our research leadership group guides our scientific decision-making. They are experts in scientific specialties relevant to Alzheimer's who lead labs at top academic research institutions around the globe. They determine what investigations we will support, always ensuring we pursue the ideas that science and data tell us are the most likely to make a real difference for current and potential Alzheimer's patients. Thank you again for joining us to hear about the science and progress that matter in Alzheimer's. We hope you will continue to join us in our mission to end this disease.
I'm really happy to be here with you today, and I'm looking forward to sharing how CureAlz is making an impact for people living with Alzheimer's disease through our funding. So my plan for today was to start off with a little bit of basics, kind of an Alzheimer's 101, before I share some of the latest research that we're excited about. So hopefully you're ready for that. So pathologists look for 3 hallmarks to confirm an Alzheimer's diagnosis in the brains of people after they've passed. The first hallmark are plaques that are made up of the amyloid beta peptide that form around neurons. Second are the so-called tangles of the protein tau that form inside of neurons, and these are the black staining that you can see in the center image on this slide. And then the third hallmark at late stages is an obvious shrinkage of brain tissue, or what we call brain atrophy, particularly in the cortex and the hippocampus, that's caused in large part by the death and the removal from the brain of synapses and neurons. These 3 hallmarks together make up the ATN framework, the amyloid tau neurodegeneration framework, that biologically defines Alzheimer's by changes in the brain. Now, these pathologies are not only common to all Alzheimer's disease patients, but they're also we also know that they occur in the order that's shown here now. That is, that amyloid builds up before tau tangles become obvious, which in turn precedes the brain atrophy or the neurodegeneration. Now, there is one other very common pathology that I've put on this slide here that's associated with Alzheimer's disease. It's gaining a lot of attention right now, and I'm going to tell you even more about this later. And I'm talking about inflammation in the brain. So inflammation can be triggered by amyloid or tau pathology, but it also might contribute to making these pathologies even worse, kind of causing like a vicious cycle of these negative changes that feed off of each other. Those hallmarks are what pathologists can see in the brain after death, and most patients would have been in very late stages of the disease with severe memory and other clinical symptoms already obvious. So an important question for which the answer helps both doctors and researchers is, how do these pathologies or these brain changes relate to the onset and the progression of symptoms like the loss of normal memory? So to answer this question, we need ways to measure brain pathologies in living people. And for this, we rely on biological markers that you'll probably have heard this term before, but we generally call them biomarkers for short. The Alzheimer's field has seen actually quite remarkable rapid advances in the past decade in the space or the field of biomarker discovery and development. There have been better and better tools that are becoming available to measure brain amyloid and tau using imaging and fluid samples. And these fluids are things like the cerebrospinal fluid, that's also called CSF, that bathes the brain and the spinal cord. As many of you know, Alzheimer's can be a very long disease. It can span decades. And based in large part on the availability of biomarkers, we now know that there is a really extended preclinical phase when people don't have obvious symptoms, but when amyloid beta levels and early signs of pathological tau are already rising in their brains. These are the first three curved lines that are shown on the graph here. There are then changes in brain volume in certain parts of the brain that start to become obvious next. And then just before obvious clinical symptoms, that's shown by the green lines in this graph, appear, we now know that tau tangles can be observed in multiple brain regions as well, although that particular line is not on this graph. All of these events, so the rising amyloid levels, the formation of tau pathology, this happens before clinical symptoms are easily diagnosed by a neurologist. So being able to measure these changes in the brain with biomarkers is really helping with earlier diagnosis and our ability to monitor responses to potential therapies in the clinic. I also wanted to say that the first biomarkers were mostly based on brain imaging using PET tracers or by measuring levels of different proteins in CSF samples. And the CSF samples require people to get a spinal tap. But we're now seeing a move to even more accurate blood-based tests that will predict brain amyloid and tau levels. And that's going to be, I think, another big game changer for the field. Some tests are already available for clinical use and others are coming soon, but all of these blood tests, as I just said, are still relatively new and they really need some further validation in real-world settings. I also wanted to add that although it's trailing behind amyloid and tau biomarkers, new biomarkers for both neurodegeneration and brain inflammation are also in development, and we look forward to seeing some results for those biomarkers in the next few years as well. So there are other important clues other than the biomarkers about how a healthy brain ends up a diseased brain. And this comes from studies of the genetics behind Alzheimer's disease. So from a genetic standpoint, there are two forms of Alzheimer's. One form is dominantly inherited early onset Alzheimer's that we also call familial Alzheimer's disease. Now, these cases are rare. They represent less than 5% of all people living with Alzheimer's. They're caused by inheriting a copy of a gene with a mutation that changes how much amyloid beta is made from its parent protein that's called APP. Patients with these familial forms of AD start to develop symptoms before age 65 and some even as young as 30. The second and more common form are called late onset or sporadic cases. In sporadic AD, genetics don't cause or definitively determine if someone will get Alzheimer's disease. Instead, it's variations in DNA sequences that code for different genes that are associated with your risk for developing Alzheimer's. We currently know of more than 30 different genes where variations in the DNA sequence can increase one's risk. The most famous risk gene is ApoE4, at least in white populations of European descent. I do think it's important to emphasize that the amount of risk conferred by ApoE4 is now known to be different across different genetic ancestries, although we don't fully understand the reason behind this yet, and it's something that we're very keen to understand. Quite a few of the known risk genes are made by the brain's resident immune cells, the microglia and astrocytes, which is another reason why there's been a lot of interest in how inflammation and the immune system contributes to Alzheimer's. Overall, genetics has taught us that some changes in amyloid beta-related genes can cause familial Alzheimer's disease, and that discovery led to the prevailing kind of amyloid cascade hypothesis that is true or applies for both forms, the early onset and late onset and familial and sporadic forms of Alzheimer's. We also know now, though, that genes are only part of the risk equation for late onset sporadic cases of Alzheimer's, at least. Many other factors contribute to someone's risk of developing Alzheimer's disease. The biggest risk factor is actually so fundamental that we sometimes even forget to say it, but it's age. This is a disease of aging. There's still a lot of unknowns about how aging processes contribute to the onset of Alzheimer's, but and that's something that, again, is another area that we're very interested in trying to identify the best research that can add some important insights into how aging is causing or contributing to Alzheimer's. But there's a long list of other factors that impact risk beyond genes and age. And some of these risk factors, which are all shown here, and I won't read through all of them, some of them, and definitely not all of them, are changeable or can be modified by personal lifestyle decisions or government or civic policies. some research suggests that addressing these risk factors for those where they can be modified, could prevent or delay up to 40% of dementia cases, including Alzheimer's disease. So it is, I think, really important to think about prevention and to understand what are the things that we can do early in life to stop these Alzheimer's cases from kind of forming at all. I think it's also important to point out here that some studies are suggesting that addressing these risk factors, things like midlife obesity and exercise, for example, can still lower your risk of dementia, even for people that have a higher genetic risk. So while you can't change the genes that you've inherited, you may be able to influence the effect that they have on your brain by addressing some of these other factors that you can change. And all signs really point to the idea that fighting Alzheimer's does not have to wait for therapies. So I'm going to end that kind of short 101 overview with what I hope is some optimism. Hope and optimism. I used a double adjective in there to try and convey to you that I do actually find myself, along with others in this field, that to be in a much more hopeful frame of mind compared to any time over the past 25 years. And there are a couple of reasons for that. First, because we've seen rapid and useful advances in the better in diagnostic and the biomarker tools that I mentioned earlier. We're also starting to see those blood-based tests for amyloid beta, which are going to be more easily accessible to a lot of people than a PET scan or a spinal tap. We've also seen positive results from 3 anti-amyloid immunotherapies now as they've made their way through clinical trials. These are antibodies that target and remove amyloid beta from the brain. Leqembi is the only one with full FDA approval right now, and donanemab is a similar drug from Eli Lilly that's under review by the FDA right now. I'd note just 3 final things, though, here. First, Leqembi was most effective in patients at the earliest stages of disease, those with mild cognitive impairment, highlighting how important these tools for early diagnosis already are and will continue to be. The second thing is that all of these antibodies can cause a rare but potentially serious adverse event called ARIA, or bleeding and swelling in the brain. Although it usually resolves on its own, the FDA advises regular monitoring for ARIA through multiple MRIs, which can be both expensive and, of course, inconvenient for patients. It's clear that that ARIA occurs more often in patients with ApoE4, so it may also not be the right choice for all patients. So we can still do better for a larger group of people. The third thing is that Leqembi and donanemab treatments both slowed down, but it did not reverse. They don't reverse or stop cognitive decline. It's about a 27% slowing, and that slowing we think, and many people think, is likely to be meaningful for patients and loved ones, but it's clearly not even close to enough. So we're not done with Alzheimer's drugs, even with these promising new treatments on the horizon. But hopefully, this is really sort of the first crack in the floodgates of new therapies that will follow these ones. So how do we get to these better future drugs? At Cure Alzheimer's Fund, we believe that research is the path to the cure, and that we'll get there by funding research with the highest probability of preventing, slowing, or reversing Alzheimer's disease. With incredibly generous donations, we have so far funded a remarkable number of grants, 850 actually, counting 2 that are not yet on the website as of today. And this has now brought us to a total of over $200 million that we've been able to fund across our 20 years in existence. We are a unique funding partner for scientists in large part because of the guiding principles that are set by our founders. We have 5 key tenets that are shown in this beautiful visual here, which is also featured in our 2023 annual report that will be published next month. So the 5 here, let me just step you kind of quickly through them. So we fund rigorous science at early stages. That's considered high risk, high reward science. We require and facilitate collaborations between our funded scientists. We're always looking for great people, promising and established scientists, I should say, with strong track records of getting work done. We also maintain rolling deadlines, which allows us to stay nimble and to be able to act with some speed to fund ideas that are really at the bleeding edge of research. And then our 5th one is that we rely on scientists as our expert advisors to guide our decisions about strategy and the specific projects that we fund. So to expand on these principles for just another minute, I divided them here into what we fund and how we fund. So what we fund includes a diverse research portfolio that covers many of the complex mechanisms that we think contribute to Alzheimer's. And this was 114 projects last year in 2023. We also fund, as I said already, great people and promising ideas at the earliest stages of research. And last year we funded 147 different investigators. We're also looking for looking to help researchers with funding that allows them to test their new ideas, maybe their wild, big, bold ideas that, and to kind of establish the feasibility or the truth behind it that will then help them to leverage that work into larger grants and investments. So we think of our funding often as really helping at that seed stage to test new ideas that other institutions wouldn't necessarily fund. We're always looking to fund research that will benefit the entire field over funding the development of proprietary molecules or therapeutics. And we require and facilitate these collaborations and the sharing of data and knowledge between our funded investigators. So how we fund in terms of speed and expert advisors here is that how we fund includes convening this world-class group, our research leadership group, which is about 40 different scientists who provide us with their expertise in evaluating projects. We also have a scientific advisory board with people who have expertise in drug development who oversee the kind of impact of our entire portfolio. We provide awards of about $200,000 per year for 2 years, which is generally considered appropriate and even generous by our scientists for this kind of seed level funding. And another hallmark of our approach is that we try to maintain a really high level of personal engagement with the funded investigators. So they do know me and the team quite well. And as I said on the last slide, we also accept proposals on a rolling basis so that investigators can apply when the time is really right for that project and not based on kind of artificial or preset deadlines. So I think by now you know we're mostly focused on funding early stage research that we also call foundational or translational research, and we're funding the research that we think will produce the novel discoveries that will get us to those new drugs. In 2023, most of our awards went to projects at this early stage, about 94% of them. But under the right circumstances, we will consider funding for research that's closer to the end of the pipeline for preclinical and clinical drug development work. Those kinds of studies can get very expensive, though, and that stage is traditionally supported by big pharma or bigger grants from the NIH. We really feel our niche in this overall ecosystem is earlier in this pipeline. But it's clearly important for us to figure out how to work together with our scientists and with other funders to make sure that academic research is able to bridge that gap. Under this early stage research umbrella, we're funding projects that fall into one of about 10 different specific interest areas. And those include some of the things that I touched on earlier, like genetic risk factors, biomarkers, amyloid and tau, as well as the immune system and the role of neuroinflammation in Alzheimer's. Several of these topics are still kind of fairly opaque or not well understood, and we do think that progress will happen faster if more heads are put together on them. So to encourage effective collaborations on these kind of even bigger, thornier questions, we also fund 5 consortia right now. Each consortia consists of 5 to 6 investigators who have agreed to work together to attack these big questions in the field. And I actually just got back from an amazing in-person meeting that we hosted in Gloucester, just north of Boston last week, where we brought together our consortia. We brought together the PIs as well as junior scientists in their labs and our entire research leadership group of advisors. It was such a good experience. There were lots of deep discussions. There was also a lot of obvious generosity on display. People were talking about sharing data and tools and seeding and talking about kind of new collaborations that they would like to work on together. So this is just to highlight that, like how we kind of facilitate bringing people together, and it was remarkable to actually see it in action too this year. Our longest running consortium that we've now funded for 6 years is the APOE consortium that's supported by the Fleming Foundation. Now, the researchers in this consortium are trying to address how exactly APOE4 contributes to risk for Alzheimer's disease. We also have an established consortium on neuroimmune interactions, where the investigators are asking how immune cells in the body and the brain are working together to maintain brain health and how these communication might go wrong in Alzheimer's disease. And then our third consortium is the Brain Entry and Exit Consortium. These teams are mapping out the brain's so-called plumbing systems and asking how dysfunction in these might cause the buildup of amyloid plaques in the brain when it should be flushed out, or how dysfunction could lead to the loss of the brain's ability to control kind of what gets into the brain. One of our newer consortium in its second year looks at the role of tau specifically in Alzheimer's. So tau is involved in other brain diseases as well, but in those cases, it's the first pathology to appear. So these are called primary tau pathologies or tauopathies. As you heard earlier, though, in Alzheimer's disease, tau is really a secondary pathology as it follows the buildup of amyloid beta. But we still have a lot to learn about what triggers that tau pathology in Alzheimer's disease and how what role amyloid beta has in triggering that. So that the AD Tau Consortium is really trying to make some headway into those kinds of questions. And then we also finally have a brand new consortium that we just convened last fall on the role of the microbiome in Alzheimer's disease. They're following up on some interesting results that show that changes in the gut microbiome can impact amyloid pathology in the brain. And they're now asking from slightly different lenses how this happens and what cell types are involved in kind of those signals that need to go from the gut to the brain. So one exciting new theme that has really captured a ton of attention at our annual meeting, I already mentioned it to you earlier, and is also evident across a number of projects in our funded portfolio, is this idea that the immune response is a key contributor to Alzheimer's disease. I'm going to give you a very broad summary of the incredibly complex immune system, just to try to get us onto the same page. So the immune system responds to infections or injuries in two ways. The first line of defense is called the innate immune response, and this response is carried out by cells called macrophages that can quickly engulf and clear away any invaders or dead tissue or anything that's not supposed to be in the body. The second response is called the adaptive immune system. Now, this is the response, or this is the part where other types of immune cells learn to recognize specific pathogens so that they can then train other cells to be kind of ready for a full-blown targeted attack if those same pathogens show up again. And this is the response that's elicited by vaccines, for example. Now, under healthy conditions, on the left of this image here, these different types of cells in the brain layer is in gray on the left, and different types of cells just outside the brain patrol and respond as needed to whatever that they encounter. Now, the peripheral immune cells include those macrophages of the innate system, and the B cells and T cells are the main cell types of the adaptive immune system. Now, the brain itself only has innate immune cells, the astrocytes and microglia. So their job generally is just kind of non-specific engulfment and clearing of foreign things in the brain. Not too long ago, we were all taught that the brain was protected from the body's immune response, and that these innate immune cells, the glia in the brain, kind of took care of anything that actually happened in the brain, and that brain again was protected from the body's immune response. And while this is true to a certain extent, we know a lot more now about how the brain and the body's immune systems are talking to each other, and we're really starting to learn how this might impact brain diseases. You should not just take my word for it, I guess, that understanding the immune response in Alzheimer's we think will be transformational to the field. We do have some evidence to back that up. This year, I thought I'd take a moment to tell you that we introduced a new, that also gets to this point in a minute, we introduced a new award from Cure Alzheimer's Fund called the Jeffrey L. Morby Prize. Now, this award was named to honor our late co-founder, Jeff Morby. He was truly an advocate for this idea that research is the only path to a cure, and he was very dedicated to finding the best ways to support scientists as they did all of this important work. Now, the award itself is meant to recognize a recent publication that's chosen by the scientific community and that's expected to transform our fundamental understanding of Alzheimer's disease and drive those insights that will lead to the new ways to prevent, diagnose, or treat Alzheimer's. So this, as I said, the winning paper was chosen by scientists as kind of the most likely to succeed, although we know it can take many years to see real-world evidence of impact of any particular scientific paper. So the winner was a paper by Dr. David Holtzman and his mentee, Dr. Chen at Washington University in St. Louis, for their paper titled Microglia-Mediated T-Cell Infiltration Drives Neurodegeneration and Tauopathy. And they published this paper in the scientific journal Nature in March of 2023. So just to support my claim that the immune response in AD is really kind of the hot topic, I wanted to highlight that as also selected by a large group of scientists, that this really seems to be an area that many people see a lot of promise in pursuing right now. So I know the title of that paper was a mouthful and full of jargon, although maybe some of the words were familiar to you after the couple slides that I just showed you. So I thought I'd give you just the TLDR version here, is that this team showed that in brains with tau pathology, the adaptive immune cells, those T cells, from the periphery actually enter the brain. They home in on cells that are carrying tau pathology, and they coordinate with the microglia, the innate immune cells in the brain, to cause degeneration of brain tissue. So these results we think are really, truly important. It opens the door to many other questions that could reveal specific molecules and signals that are used by those cells and for which drugs might be able to target and then stop those processes. It also helps to drive a whole other list of questions in the field that I'm showing you here, including things like, are there fluid biomarkers that can tell us about a person's neuroinflammatory state? Can we harness the adaptive immune response to make newer, safer therapies? And then there are a couple other interesting ones in here too. I won't read all of them to you, but I do want to say one that we're interested in is, why do vaccines broadly appear to reduce the risk of dementia or Alzheimer's disease? So there's something there about the peripheral immune system that might be mediating that protection that people are observing with vaccine use. So at Curals, we really do think that funding this early stage discovery-based studies on the immune response, as well as many other areas that I didn't go into any detail today, will push the field to discover that new biology that will get us to those new drugs. So we feel that one of our roles is to really help kind of fill the hopper of the drug development pipeline. We want to help fill this with targets that have strong scientific evidence, and then see them, go through rigorous randomized clinical trials that will prove whether or not they're actually effective in patients. Quite a few drugs are working their way through clinical trials right now. This is a bullseye diagram from that's prepared every year by Dr. Jeff Cummings at UNLV, and it shows all the drugs that are targeting Alzheimer's that are in either phase 1, phase 2, or phase 3 clinical trials. And so phase 1 trials are those in the outer ring, phase 2 trials in the middle, and phase 3 trials in the center. As of January 1st this year, there were 164 active trials with 127 drugs that are being tested. 32 of those are already in phase 3, so the closest to kind of completion of a trial. Oops. And most of those drugs are what we hope will be disease-modifying therapies, but meaning that they will actually either, again, slow the actual progression or potentially even stop the progression of the disease. But the orange and the blue pieces of this pie here are drugs that are actually targeting symptoms. And so there's still a lot of effort in the field to identify drugs that might help with cognition or might help with other behavioral symptoms as well. Now, the target of these drugs and how they work are actually pretty diverse. It used to be that all the drugs on this chart targeted something to do with amyloid beta, which are the symbols in red. But now we see many other mechanisms, including inflammation, which are the symbols in yellow, and tau, which are in blue. So I do think that it's a time for optimism and hope in the AD field, but there's no question that we still have a very long way to go.